I will describe the experience of applied stem cell therapeutics in second-generation gene therapy and gene editing for hemophilia A. Next slide, please. I will briefly outline the state-of-the-art therapies for hemophilia A and the key components of our second-generation gene therapy for hemophilia A. I will explain the advantages of using in vivo gene editing using CRISPR-Cas9 for hemophilia A, and you will be able to learn about unique manufacturing and clinical aspects of gene therapy. Next slide. Let's take a scientific journey from the lab to creating a biofactory in the hepatocytes of patients with hemophilia A. Next slide. Nowadays, we have several effective therapies, mostly based on recombinant factor VIII replacement. And if Roctavian, a first-generation gene therapy, is approved by the FDA this week, we will initiate a revolution in the prevention and treatment of hemophilia A. However, while gene therapy can become the treatment of choice for adult patients while eliminating the need for prophylactic replacement therapy, the pediatric population requires gene editing to fully integrate the replacement gene into the cell. Next slide. The initial research on gene therapy for hemophilia A started in the early 2000s. In contrast to other gene therapies, it was quickly realized that the transfer of the native functional gene to replace a defective factor gene was insufficient to achieve therapeutic factor VIII levels. This timeline shows when several biopharmas initiated or will initiate their gene therapy clinical trials. The key takeaway is that once all decisions are made regarding a factor VIII construct, it's impossible to get back. The train has already left the station. I will highlight today how that scientific progress and technology doesn't stand still, and improved therapeutics are always building on previous experience, especially relevant for the second-generation therapy for hemophilia A that I will be describing today. Next slide. While factor VIII can be synthesized by several tissues and in different cells within the liver, the hepatocyte is considered the perfect target as a biofactory. To the left, we have the main element of a gene therapy infusion, a viral capsid containing the factor VIII transgene. To the right, we see how the virus, any virus by the way, enters the hepatocyte, is captured by the endosome, and transported to the nucleus as a circular strand of DNA. Note that the circular strand of viral DNA containing the factor VIII construct is not inserted in the chromosome, which are in a green color. This is called an episomal location. This lack of integration explains why factor VIII gene therapy only works in the adult population. For our clinical trials, 18 years or older patients. In younger patients, the gradual turnover of hepatocytes leads to the loss of these episomal viral genomes. Next slide. To understand the notion of a second-generation gene therapy, let's dive into the assembly line of the hepatocyte biofactory. And this is in some way a bit of high school biology. A second-generation gene construct will improve biosynthesis and secretion of factor VIII and ultimately achieve long-term expression and require lower doses of vector genomes, reducing costs and immunogenicity. Refining the factor VIII construct is critical because native human factor VIII is poorly expressed naturally due to inefficient biosynthesis and secretion. In the upper table, we see that all first-generation factor VIII gene constructs currently in development include a liver-specific promoter, a B-domain deletion that allows packaging into the virus, and an optimization of the DNA bases or codons to improve transcription, which is the way the DNA transcribes into RNA. For example, a decision has to be made at some point if CAA or CAG is going to be used for most efficient coding for glutamine, and the same for other amino acids. ASC 618 is a second-generation construct, which includes all the characteristics of a first-generation construct that I just described, with a more efficient liver-specific promoter, and more importantly, a hybrid human porcine sequence of factor VIII that enhances the translation of the linear amino acids into the therapeutic factor VIII protein. This leads to an optimized post-translational folding. In some way, this approach minimizes the intracellular retention of unfolded factor VIII in endoplasmic reticulum and Golgi apparatus. Again, this is high school biology, but the key is that first generation is focusing on transcription, second generation is focusing on translation from the linear amino acids into a protein. Next one. We have licensed our factor VIII construct from the group of Professor During and colleagues at the Emory University. They have been studying the phylogenetic trees of factor VIII and identified specific sequences that increase post-translational efficiency, as shown in the previous slide. So this is a recombinant hybrid human porcine factor VIII that incorporates selective porcine sequences necessary for high-level expression driven by enhanced biosynthesis and secretion. Next slide. To describe the gene therapy for hemophilia A, I'm using as analogy the Apollo 11 historic journey to land a man in the moon. It required a rocket with a propulsion capsule coupled with the lunar module. For gene therapy, we also need a rocket, the viral vector, which carries a payload consisting of a promoter, the propulsion capsule, and a transgene, the lunar module. Next slide. I know the audience has nowadays a significant knowledge about viruses, especially the one at the bottom right. In hemophilia is the adeno-associated virus, AAV, also a spiky virus, but completely harmless for humans. It has a navigation system that preferentially reaches the liver. Next slide. Our factor VIII construct, ASC 618, has two key elements, the promoter, like a propulsion capsule, and the transgene, like the lunar module for the Apollo 11. The promoter activates the expression of the transgene of the factor VIII, and the protein structure of the entire cassette is shown here. This conformation is different from the first generation factor VIII proteins because it has been optimized for increased biosynthesis and secretion of factor VIII by the hepatocyte. You can see here 91% of the sequence is human, 9% is porcine. Next slide. In this line graph, we show that the promoter and the transgene of ASC 618, which is the upper line, when injected intravenously in mice, are more efficiently biosynthesized and secreted than first-generation factor VIII constructs, which are in the lower line. And this is publications by the group of Emory. In the next slide, we have confirmed internally in our work in our labs at ASC Therapeutics that as following the requirements of the pre-IND meeting with the agency, the FDA requires preclinical animal studies to be conducted in 3 different animal species. And we have done that in wild-type mice, humanized mice incorporating human liver cells, and non-human primates. Our results show that the factor VIII protein levels are significantly elevated in a dose-dependent manner, that factor VIII protein inhibitors are dependent on factor VIII protein levels, indicating successful factor VIII protein production following ASC 618 treatment, and there were no adverse events observed in non-human primates, including body weight, body temperature, clinical signs, liver enzymes, and hematological parameters. Next slide. In summary, our gene therapy program had an extensive discovery program to create the viral vector construct with second-generation promoter and a hybrid transgene sequence. And all this has been previously published. And we can see here one of the key publications of that team. Next slide. Let's now outline our gene editing approach to insert the factor VIII transgene into the genome. Next slide. ASC 518 gene editing is a separate clinical program that uses the same bioengineered hybrid transgene for factor VIII, but this time integrating it into the genome. It's like landing the lunar module on the surface of the moon. Being able to integrate factor VIII into the chromosomes means that factor VIII production continues despite cell division of the hepatocyte. Typical hepatocyte turnover observed in the pediatric population. Our research team has developed a CRISPR-Cas9-based gene editing technology published in Nature Scientific Reports using two steps. First, we inject the viral vector AAV8 containing the CRISPR-Cas9 enzyme with a guide RNA identifying a unique DNA sequence adjoining to the albumin locus in the hepatocyte. It makes a precise cut like genetic scissors. Next, we inject another viral vector AAV8 containing our second-generation hybrid transgene, but without the promoter. We have demonstrated in this publication that the promoter of the albumin locus activates the transgene to synthesize and secrete factor VIII in Hemophilia A animal models. Next slide. These are the advantages of gene editing. Lifetime effects independently of hepatocyte turnover, which is especially high in the pediatric population, as I mentioned before. In contrast to gene therapy, gene editing allows the integration of the transgene in the genome and is always available. Also, high potency and efficacy, taking advantage of the highly active albumin promoter, and safety. We see a site-specific integration in the genome. Next slide. Gene therapy also requires unique expertise and coordination for manufacturing and clinical studies, like the Houston's mission control for Apollo 11. Next slide. For those of us that have been in clinical development for some time, we have experienced a correlation of the molecular size of the therapeutic agent with the manufacturing complexity. I started my clinical development career with a biologic, a Humira, about now 20 years ago, and we felt at the cutting edge, like piloting a jet fighter. From the time I joined Avexis clinical team developing the gene therapy Zolgensma, working on Hemophilia B gene therapy for Unicure, to my current role, I feel like an astronaut in a space station. Next slide. Every biopharma needs to develop sophisticated processes to facilitate manufacturing and ensure efficient packaging and transduction of AAV. For ASC 618, we introduced 3 DNA sequences or plasmids into producer cells called HAK293, which then deliver the recombinant viral vector with the integrated promoter and transgene. When this recipe is cooked in the bioreactors, we obtain a supernatant and a pellet of cells. Both contain the viruses and require fine-tuning to optimize the yield of bioengineered genes. As Chief Medical Officer, I must understand this workflow of viral gene delivery to ensure it meets my needs in terms of viral gene copies for my clinical trials. The analytical methods to measure viral titers and viral genomes in the final drug product are going to inform the methods I use for my gene therapy clinical programs. Next slide. And finally, I just want to give you a sense about the way we conduct our clinical studies. There are critical elements of our Hemophilia A gene therapy clinical programs. We're selecting a severe population of patients who will benefit the most from a single infusion that will actually cure their disease. a single infusion that will change their lives. No more factor VIII replacement therapy or treatments for bleeding episodes. Besides the standard phase clinical program, the FDA requires us to follow the patients for a minimum of 5 years. The unique safety endpoints of a gene therapy study include assessing the AAV infectivity in the liver and other organs, and assessing the immunogenicity of AAV to factor VIII. For efficacy, the FDA wants to see factor VIII activity in blood and changes in bleeding episodes and replacement therapy. I hope you now will see viruses with different lenses and as rockets powering a new era of genomic medicine. Thank you so much. Last slide.